Okay, so we just went over fats, phospholipids. You should be able to identify those, understand the importance of hydrophilic, hydrophobic. We went over a little bit about cholesterol, the importance of fat in our diet, steroids, different things like that. So now we're going to talk about proteins. And protein is the machinery in the cell. Proteins are what do the work. It does everything. And it's all about the shape when it comes to proteins. If the shape of the protein changes, all of a sudden it can't do it. And think of it this way, if you got a shovel, it has a certain shape to it to help dig. Well, if you change that shape, if you take and bend that shovel head into a rose, is it going to dig very well? Well, absolutely not. So think of that with proteins. It's all about the shape. And so they are involved in nearly every dynamic function of your body. So they're just extremely important. Very diverse, all sorts of different shapes, and the specific structure determines its functions. And they're all just kind of different arrangements of 20 amino acid monomers. So amino acids, they have an amino group and a carboxyl group, which makes it an acid. Bonded to the center carbon is a hydrogen atom. And so that's symbolized, the chemical group symbolized by an R, which determines the specific properties of the 20 amino acids used to make the proteins. So an amine group, carboxyl group, and there's your R. There's your drawing. And so they're either classified as hydrophobic or hydrophilic. They either like water or they hate water. Phobic, hate, like, philic, or love, whatever. So here's a hydrophobic amino acid or protein and hydrophilic protein. So the amino acid monomers, they're linked together once again by dehydration reaction, and that's what creates what they call a peptide bond. And that peptide bond will be important when we talk about the formation of protein from DNA. And so you can take that and join one monomer and make another and create this huge change of amino acids. When you get a chain of amino acids, it's called a polypeptide. Poly, multiple. They claim one of the most important roles for protein is as an enzyme. I don't disagree, but just know that enzymes they lower the energy of activation, and they're part of almost every single chemical reaction in our body. And so proteins, they serve as metabolic catalysts. They get things going, and they regulate. They stop them before they go too far. So start and stop. Think of it as the gas pedal and the brake pedal on your car. They're kind of one of the most important parts of that vehicle. Sure, the steering wheel is important, but only important if you're going or stopping. Here's some proteins do other important things. They're structural, contractile, defensive. They signal the receptors, transport, storage. Proteins do everything. It is all about the proteins. I've done a lot of climbing, and this doesn't look like a really highly trained climber. And I gotta say, I think that's probably photoshopped. Any good climber knows that you keep the weight on your bones, not on your muscle. Polypeptide chain contains hundreds or thousands of amino acids. And so it's the sequence of those peptide bonds that make a particular shape. And it's the shape that determines its function. Think of that shovel, okay? So here's a protein. It shows the groove site. The shape of that groove site is what's going to cause or stop reactions from taking place. And so here's a 3D molecule, and you have that groove, and it's a very specific groove, and anything that doesn't fit in there, any compound that doesn't fit won't work. So if we change the protein shape, it cannot function. Right? The process of losing a shape is called denaturation. When it denatures, the nature of it has been removed, the nature of that shape. And so as soon as it loses its shape, it has lost its function. And so this is real important why fevers are important to watch, because if you get too high, if your fever gets too high, you start to denature the proteins. So what your body's doing with the fever is trying to elevate your temperature enough to denature the proteins in the bacteria that's causing the sickness, but not so much as to denature your own proteins and not hurt yourself. And so it's kind of a tough balance sometimes. And so proteins, they can be denatured by changes in salt concentration, by pH, either acid or base, or by high heat. So it's important that your proteins, if you want to keep them functioning, you avoid those things. So proteins have all sorts of different levels of structure. Primary, secondary, tertiary, quaternary. 1, 2, 3, 4. Those are the structure levels. Primary structure is when you're making those peptide bonds and they come together in a chain and you have that polypeptide. Okay? That is the primary structure. The slightest change in that sequence of the polypeptide will affect how the protein folds and will affect its ability to function. So the second is when it starts coiling or folding. So when it coils up, it results in a helical structure, or when it folds, think of kind of like a pleat. And so you get that kind of pleated sheet, and so those are secondary structures. And so spider webs are proteins that they use. And then when it folds in on itself, so when you get those helical structures and those pleats, when they start to fold in on themselves, they're tertiary structures. And those result from the different functional groups. And then you get disulfide bridges. And then when you take two or more proteins that folded in on themselves and put them together, you get a quaternary structure. So the common one is hemoglobin, like we saw at the beginning, a quaternary structure, 4 different proteins all coming together. They have this polypeptide chain that then folded in on itself and made helical structures and pleats, and then that folded in and made a 3D structure, and then those came together with other proteins and made a quaternary structure. In this case, they're using collagen in your bones, and so it's a triple helix and gives it great strength for connective tissue. And so here we go. There's your 4 levels. Primary structure at the top, it's amino. Each one of those is an amino acid. So there's your polypeptide chain. Then when it starts to make pleats, pleats over there on the right, or the alpha helix on the left, that's the secondary structure. And then when it folds in and makes a 3D structure, that is a tertiary. And when you add one or more of those together, then you get a quaternary structure. So those are a brief introduction to proteins. Answer the question, we'll go through nucleic acids.